today's topic is protein for crystallography. Chapter title is like that, but I'd like to replace that crystallography for protein. Anyway, we are talking about protein as well as crystallography. So what do you have in your mind in terms of crystallography now? Anyway, we talked about crystallography at the beginning. Anyway, we talked about cell lysis, but what about crystallography as your mental image? We are talking about proteomics. We should to get the sample, we should do cell lysis first. Otherwise, we cannot analyze proteome inside of the cell. In such a way, for example, cell lysis for proteomics. So crystallization for proteomics, not proteomics, crystallization for protein for crystallization. So what I'm trying to say is here the title is protein for crystallography, but in reality this book means protein for crystallization. Okay. That is the message I'd like to share. Then, and why do we need protein for crystallography? First of all, why do we need protein? It's a kind of open question.
I think in this part, it mentioned about the protein. We need to prepare protein to get the structure, protein structure, and
yes, structure.
and
That's the point I'd like to share. Yeah.
and more than structure, we can research about the function of protein.
Function. Structure, function. That's the keyword I'd like to hear from you. So, unlike proteomics, protein crystallography is focusing on each protein at each experiment. Protein analysis covers whole proteins theoretically, not in practice. In practice, you may cover several thousand, but total gene expression may result in 30,000 for human, but we may cover only 10% or less. But protein crystallography is dealing with one protein at each time. As Dr. Mui said, to know the structure for its function, or even we can change the structure, not change the structure, we can change some amino acid inside the protein and we can modify its function. That's the purpose. So, purpose is to study structure of protein for crystallography. So, I'd like to remind you the purpose. So, with purpose, we will go through protein engineering needed for crystal. Actually, we will not cover protein engineering in total, but today I'd like to cover some vocabulary. I'd like to share some vocabulary or language aspect with you, and you yourself can study for yourself, can study the topics if you need the details. But we cannot cover details through this coursework, but I'd like to help you to develop the skill to study for yourself. That is objective of this study coursework. So, to study protein structure, normally we are not using protein expressed in the original cell. Then what we are going to do? Why it is needed? Sabah, for your own benefit, I talked about proteomic analysis, not crystallographic analysis. Proteomic analysis, you are growing chlorella cell, and you are analyzing proteins in the chlorella cell. But we have difference between proteomic analysis and crystallographic analysis. Even for chlorella proteins, we are not growing chlorella cell to obtain such a target protein. What we are doing for crystallography?
We clone the genes and then put it into the other strain of host and then grow it, and after that obtain it and purify it.
Yes, exactly. Clone is the keyword.
Yeah.
Clone is making something identical in other place. So like a twin.
Yeah.
So why do we need cloning, first of all?
Cloning, because we want to have a large amount of.
A larger amount.
Yeah. For example, when I get the target gene and I clone into E. coli or some other bacteria, because E. coli or bacteria grow very fast.
Fast.
And we can get a lot protein we want. Yeah. And we can save time.
Save time. Very critical point. Yeah.
Save time and we get our purpose.
Not only saving time but also saving money in terms of culture media.
Yeah.
For example, human cells are difficult to grow. Not only difficult to grow in terms of time and effort, but also it's expensive in terms of culture media. So, chlorella, plant, human cells are animal, or animal cells, even animal cells are not easy to grow like bacterial cell. So, it's better to use another kind of host like bacteria. Simplest one and most well studied one is E. coli, as you said. So we normally use E. coli as a first choice. But unless E. coli is to express the target protein, we may use another one, like CHO cell for human cell expression. But normally for chlorella, we may be able to express any chlorella protein inside of E. coli without very difficulties. That's the point. We are using recombinant DNA technique to express target protein in foreign cell to original one. Okay, so here, what is subcloning? We talked about cloning. Cloning is selection of host cell and expression of target protein in such a host cell. So, but here we have another level of technical words, subcloning.
Subcloning is one technique in cloning, gene cloning. We can use there are two kinds of vector. Yeah, the first one very easy to insert our target protein, target gene in the first vector, and then we use this vector and transfer into one kind of host cell. And in this host cell, this vector can grow a lot, and then we digest one more and transfer our target gene into the second vector.
Very good point. We have two different vectors,
Yeah, right.
first one and second one. So we are actually cloning from first one to second one. But unlike the word cloning, subcloning is at different level. So cloning between foreign cell and host cell. Subcloning between which cell and which cell? You are saying different vectors, but what are cells in those two cases? cells are the same.
Yeah.
We are using the same host cell.
Same
So
host.
within same host cells, we are changing vector,
Better. Just
original vector and new vector.
new better.
Why do we need new vector?
Because maybe the second vector can grow. We can get the high efficiencies.
Yes,
in
yes,
the cell.
high efficiency in terms of
Second vector.
expression.
Yeah, right.
So first one may be good for manipulation. So to have expression, first of all, Saba, I'm sorry. Did you know something about cloning and subcloning? The difference between cloning and subcloning? No. Then, and you didn't do any genetic engineering yet, or simply recombinant DNA work yet.
Yeah.
So for you, I'd like to review some key concepts. Any proteins for CRISPR is all about protein engineering for crystallography. Also, for you, we have some comparison. I'd like to make some comparison for you. Proteomics. Proteins for proteomics. Okay, you are studying this, proteins for proteomics. The reason why you used French press is to obtain proteins for your proteomic study. Likewise, we have proteins for crystallography. The purpose of proteomic analysis is mostly identify proteins inside the original cell, expressed in original cell, because we are concerned about some target cell. Here, we may have some target proteins. This is multiple inside of target cell. Target is our topic for study. Your case is cellular cell, right? But this case, in terms of amount of protein expression, we would like to see target proteins as expressed in target cell at the time of observation. That's the whole purpose of proteomic study. Likewise, we have target cell, not target cell, target protein expressed in target cell, but we are talking about host cell, not target cell. Why we are differentiate between target cell and host cell while we are talking about same target protein is like this. Purpose of proteomic study is to measure expression level or amount of target protein expressed in target cell. So we should look at target protein directly. But purpose of protein crystallography is not look at amount of or expression of target cell, structure of target cell, target protein. So to see target protein structure, not the target protein amount, here point is here amount or expression. Here, not amount but structure. Structure. If we translate amount in terms of another word, and this is copy number. How many copies or how many molecules as expressed. But here we are not concerned about copy numbers. We are concerned about enough amount of target protein to study its structure. So here, more copy number is better. In terms of trial, we may need more proteins. If we have more proteins, we can try more. So even for crystallization, we need some amount for screening. Even for structural characterization, still we need more. So more protein is better. So to get more, original target cell and original target protein expression level can be low, especially for signaling protein. For skeletal protein like actin or some muscle protein, biomass protein, high. But signaling or some control protein is very low. Enzymes can be very low. So in such a case, we need to express such a target protein in not in target cell, but in another cell, kind of cell factory, so-called factory, a cell factory or host cell. In this case, so literature search will guide us to identify our target protein. Then we should insert our target protein sequence into host cell. That can be done with at DNA level. So at the DNA level, especially for E. coli, many vectors were developed in terms of adding foreign DNA sequence into host cell. That's the word vector we are using for that purpose. Transferring or inserting foreign DNA sequences into host cell. That's the vector. So we are using the word vector to transfer target protein sequence, actually DNA, target DNA into host cell. That's the vector. So in this way, we can clone or copy, simply copy target sequence into host cell. That's the clone, and subclone is copying again. And we can have vector at the molecular biology textbook, or even in this textbook, we have some summary about DNA work, and you can find vector as well as our target sequence and circular vector. So we can insert our target sequence into such a vector, circular vector, and we can transfer such a DNA sequence, circular DNA sequence, into host cell by transfection. Here, where do we have transfection? Transfection. Thank you. So now we have some common vocabulary. Host cell and target protein expressed in target cell is like proteomics, but we are expressing target protein not in target cell but in host cell for overexpression. To do overexpression, we may need some induction mechanism using chemical like IPTG, and we can obtain some amount, enough amount for here scale up. Firstly, to check its feasibility of expression, we are testing at small scale. Then once it's confirmed, we obtain large culture. Here, some milliliter scale, and here liter scale. Then we may obtain in several gram. Then we may, I'm talking about cell pellet. Like a proteomic study, we after centrifuge, what do you obtain? Cell pellet. We obtain cell pellet. You can paraphrase this. Biomass is actually cell pellet. After cell culture, we obtain biomass, which can be regarded as cell pellet. Then lysis, and we are obtaining proteins. But here we have difference here. Capture. Proteomics. Proteomics should analyze protein amount as present in target cell, but we are not interested in whole proteins, but we are interested only target proteins. So we should capture only one at this stage. But we may capture some panels of proteins. Do you use capture or not?
No.
For example, for medical proteomics, we may capture using phospho affinity tag, and we may analyze only phosphorylated proteins. But in your case, you are analyzing whole proteins, right?
Yes.
Then you may not capture anything. We just analyze whole. Even you will not use purification, polishing, concentration. So after isolation and lysis, you are removing other part of cells.
Yeah.
Then you are just analyzing. Actually, you are digesting.
Yes.
With trypsin.
Yes.
You are digesting trypsin, and you are analyzing peptide,
Yes.
not protein.
Yes.
Peptide mixture.
Mm-hmm.
Here, but we are not analyzing mixture, but we will use purification step with capture, and we will have concentrated protein at the end. Then we will do crystallization. So in proteomics, we have mixture of proteins. In crystallography, we would like to have single protein of high purity, for example, 99%. Even after single purification step, with especially here, the word capture is used in terms of histech or any affinity chromatography. Mostly at first step of purification, we are saying capture or histech. So just with such a single step, we can achieve higher than 90% purity. So we have almost pure in terms of its appearance or color, but it may not be pure enough for crystallization. Crystallization, we need more like 99%. So to achieve such a level of purification, we may do extra step as a purification, and that is usually ion exchange and gel filtration. But you may not do now. But what I'm trying to share in terms of vocabulary is this has something common with proteomics. But proteomics, protein mixture. Crystallography, single protein. But single protein overexpressed. Proteomics, protein mixture as expressed in original cell or original target cell. So we are using same lysis step. Also, we are using same cell culture, but we have differences. Anyway, proteomics is about sequence, peptide sequencing, right?
Yes.
Using mass spectrometry. So result is sequencing. But crystallography is input is sequence. I'm trying to help you in terms of end and beginning. Proteomics, once we have mixture proteins, after trypsin digestion, we obtain mass spectrometry analysis, and the result is peptide sequence. But proteins, what we are doing is we are trying to search a sequence, target sequence. Here, mixture sequence. But here, we are putting target sequence into the cell, host cell. So here we have multiple target of interest. Here we have single target of interesting sequence. Sequence is originally DNA sequence, but we can translate such a DNA sequence into protein sequence, vice versa. If we have protein sequence, it can be translated into DNA sequence. So what we are actually putting here is DNA sequence in vector. We can synthesize DNA, or we can extract some portion of DNA from target cell. using restriction enzyme. Yeah. Then we can ligase such a portion back into some vector, and we can insert such a vector into host cell. So we have some similarity and we have some differences between proteomics analysis and crystallographic analysis. Okay. Saba, any question or comment?
No, I get it.
So you at our lab meeting, when others are talking about crystallization or protein engineering, you may have some idea or parallel comparison between proteomics and protein crystallography now.
Yeah.
That's the purpose. So conceptually, as we talked about, DNA level and protein level, and proteomics start with protein, and proteomics start with protein mixture down to sequence. But crystallography is starting with DNA level down to protein level. So what we are actually obtaining is protein structure. Sequence should be known at the beginning. Without knowing sequence, we cannot even synthesize or even inventing such a construct. Let me talk about construct a little bit. Now let's talk about model. Model of molecular biology. In molecular biology. So in crystallography, in terms of physics, we are talking about actually atoms. We are not talking about other subparticles like quark or muon or even electrons directly. We cannot see electrons directly. We may see some electron density, but we cannot identify each electron. So what we are using in terms of physics model of crystallography is atomic model. Likewise, in biology, we are using this model, DNA, RNA, and proteins in terms of sequence, each sequence. This is, I'm checking elementary knowledge of molecular biology. What is the name of this step?
Transcription.
Oh, that's good. Transcription. Transcript. And what is the name of this level?
Translation.
Oh, very nice. Translation. Transcript is like changing words. Script is words, but this changing location. This is not simple translation, but or this has some implication in space. Structure in space. Sequence is mostly linear information. We are not concerned about its shape. Any sequence information is mostly linear. So DNA, RNA is a little bit tricky because we have RNA enzyme. What is the name of RNA enzyme? This is just checking your common sense in biology.
RNAs.
RNA is one of such an enzyme. Nice. RNA is P. RNA is P. Not all RNAs may be ribozyme, but RNA enzyme is called ribozyme. Ribozyme. But for the time being, let's ignore RNA. This is a little bit tricky, but just skipping here. And in terms of information flow, DNA, if we are given DNA sequence, then we can translate to proteins. So in terms of script, we can omit this and we can translate directly down here. Okay? This is not for biology model, but this is for proteins for crystallography model. At the cell level, especially for proteomics, also we have nowadays RNA-seq, RNA-seq. That's a topic which Dr. Gang is very interested in recently, and we are talking about transcriptome. Did you hear anything about transcriptomics? Transcriptome. Transcriptome analysis, like proteomics. But for simple view, we will not talk about transcript, but we will talk about DNA sequence and protein sequence. Proteomics, DNA sequence, and peptide sequence. Okay, so we will talk about sequence of DNA and protein sequence. And as a result, we will reveal structure of proteins. Sequence and structure. So here, DNA level, DNA sequence. And protein level, we have corresponding peptide sequence or protein sequence. But we have some problems in terms of hierarchy of protein structure. We have primary structure, which is actually protein sequence, and secondary structure. What is secondary structure in protein structure? What's the name of such a secondary structure? This is a quick review of chapter 2.
Beta.
Yes, alpha helix and beta sheet. So upon them, we have some organization, tertiary structure. And domain structure can be organized into quaternary. Again, okay. So anyway, here, although it's not mentioned, DNA level and protein level, two views. We are omitting RNA level. RNA level is a little bit complex, but for simplicity, that's the purpose of I'm talking about model. Here we are not including RNA model. In our view, simplified version of molecular biology, because like dictionary, if we are given about DNA, we have corresponding protein by automation. We have just, depending on species, we have direct translation between DNA and protein. Okay. So DNA level and protein level, for DNA sequence, what we are actually having is protein sequence. But sequence, in this, I'm trying to help you in terms of reading the textbook. Textbook may distort reality, or it may not have good correspondence with reality, or it may distort some idea. So here, to be more correct or accurate, DNA level, if you have DNA level, protein level, quite symmetric, level and level, DNA and protein. Here, DNA sequence, but we do not have protein sequence. Here, as a red label, red letter DNA label. Here, protein label. Logically, we can assume DNA sequence and protein sequence, but here we do not have such a protein sequence to be symmetric. DNA label, protein label, we should have protein sequence. Okay, so here, rather than having protein sequence, we have two things: expressed protein and crystallizable protein. What is the difference between expressed protein and crystallizable protein? They are both are protein.
Purity? Like protein is expressed, but for crystallization we need pure protein.
That is one idea or suggestion. But what I'm trying to help you is decode the meaning hidden in the text, or let's be logical. Here, DNA label, protein label, DNA sequence, first of all, we are missing protein sequence. But protein sequence can be expressed as a real protein. We are talking about real protein, not just sequence. Sequence is information. But also, it can be realized into material. What I'm trying to distinguish these two things. In terms of word or vocabulary, we need to distinguish information and actual material based on information. Usually sequence, with the DNA sequence, we can order DNA material, simply DNA, with our sequence information. Sequence information and, let's say, DNA material. DNA synthesis is easy or cheap. Protein synthesis is not cheap. That's why we are using many steps of purification and many steps of cloning. But DNA as a material is simple, and also DNA as a full sequence, it's easy to synthesize. So people have tendency to use sequence as material. Here, DNA sequence can be easily converted to DNA material. That's my comment. Once we have DNA sequence, it can be easily realized into DNA material, cheap, at the price of $100, not very expensive. But protein can, even if we have protein sequence, it cannot be synthesized easily. If it can be synthesized easily, there is no point to do lengthy steps of purification and complex design. Okay, so here, let's regard DNA sequence as DNA material. Okay, both are equivalent. So, but here, expressed protein, crystal protein, also material. Expressed protein material and crystallized protein material. To be crystallizable protein, or even to get any protein in terms of material, not in terms of sequence, we should have expression. So simply, this is another version of expressed protein, or one of such expressed proteins. But what is the difference between these two? Is simply this is crystallizable, and some of expressed protein may not be crystallizable. So crystallizable and non-crystallizable, both are possible. But here, only crystallizable. So even we may not guarantee expression at all. So DNA, protein, and for the time being, we will not talk about RNA. So DNA sequence information can be easily converted to DNA material, but proteins, it's not easy. Protein, I'll talk about protein, can be classified into 3 levels. In terms of cloning. In the host cell, if we are talking about chlorella proteins, always we have some amount of chlorella proteins in some way. Depending on cell cycle, some may not be expressed, but we can say 0 copy. Such proteins can be found at different expression level. But target protein expressed in host cell, it cannot be always present. So even expression itself cannot be done easily. So we can have no expression or not expressible or expressed. Proteins in, so here, target proteins, we should say target protein in host cell. We are not talking about target proteins in target cell. That's for proteomics. But target proteins, we would like to insert some sequence information, and we would like to have such a protein as expressed in host cell, but such expression is not guaranteed. So it cannot be either expressed or not expressed. So we have not expressed case. So even if we have some expression, already it's a kind of success. That is expression test. Second level is even for expressed, we have insoluble and soluble. So from cell lysis of cell pellet, you may have supernatant and remaining some solid part. Your protein may be solved in soluble portion or insoluble portion. But for crystallization, if our expressed one resulted in insoluble one, we cannot rescue them. So we should have some soluble protein. Soluble part. Even for soluble one, now that is the hidden label there, but any expressed protein means in that case soluble, let's suppose, then even soluble one has a crystallizable and non-crystallizable. Crystallizable and no. Okay. We have this level of complexity. Target protein either can be expressed or cannot be expressed. Even if it is expressed, it can be insoluble or soluble. Among soluble, we have crystallizable and non-crystallizable. Okay. So here we will focus this side. This is for combinatorial design. You can read something about combinatorial design, but we will focus on targeted design in terms of sequence manipulation. Here, targeted mutagenesis, usually I'm doing targeted mutagenesis, especially single point amino acids change. Then we can have corresponding here DNA sequence analysis, and here we can have same DNA protein sequence analysis, but here this DNA level is talking about sequence level. Here we are talking about material level. This view is about sequence, this view is about material. So expressed protein, we can do some solubility analysis or aggregation analysis. A kind of protein can be dimerized or it can result in polymer. So protein analysis, and we can have some treatment to have crystallizable protein. Even for his tag, we can cut his tag. So his tag is for capture and kind of artifact added to target protein. Saba, do you know something about HISTAG, or did you hear something about HISTAG?
I just heard the name, like
Mm.
Mohammed keeps
Mm.
talking about it, but
Mm.
I think the protein is combined with
Mm.
HISTAG,
Mm.
is tagged with,
Mm.
I don't know
Mm.
actually.
Next slide will show you what HISTAG is and any HISTAG. Let's talk about HISTAG later. I have something for HISTAG, but let's talk about HISTAG right now. I'm sorry. HISTAG. In terms of proteins, this is ribbon structure, and his, 1 his, 2 his, 3 his, 4 his, 5 his, 6 his. In this model, this is not real protein, but this is model of protein drawn with alpha helix red and beta sheet green. Main chain, I did say main chain, linker or chain in cyan, and among such a chain, his tag was drawn with ball and stick model. Here, blue one is nitrogen and gray is carbon, and it was shown with 6 his tag, 6 his amino acid. So we have 6 his amino acid in series. I'll tell you something about the purpose of adding such a 6 his tag. It is good for affinity chromatography with nickel. What is the name of nickel? NTA color. Yes. So this portion of protein can be bound to nickel NTA column. So it's for capture. But in original protein, in nature, we never have this kind of tag. This tag is for purification purpose or capture purpose. In crystallography, it's not easy to capture single protein out of mixture of proteins. So unlike nature, we are adding such an extra amino acid at the end of either at the end of C terminal or at the end of N terminal, at the beginning of N terminal. So this is kind of artificial part. This is for purification purpose. Without such a tagging, it may not be easy to isolate one single protein out of protein mixture. Actually, what we are doing for purification or sample preparation is very like proteomics. You are growing chlorella, then you are doing cell lysis, then you are extracting protein mixtures. Likewise, we are normally in crystal, we are growing E. coli as a host cell, and we are harvesting some target protein out of E. coli cell pellet. But difference is, with the presence of such a his tag in target protein, we can easily capture or separate this target protein out of protein mixture. That is not possible for natural protein, but it's very nice to have such an artificial protein. So recombinant protein for crystallography always have it always has such a tagging protein for most times. For very abundant proteins in the early days of crystallography, we didn't have such a his tag. For example, first protein structure determined is hemoglobin and myoglobin. They are abundant from horse blood. Myoglobin, hemoglobins are proteins from blood, and any cows and horses have huge amount of hemoglobin and myoglobin from their blood, and it's easy to have such abundant proteins with simple precipitation. But for other proteins, especially target proteins in health and medicine, it's not possible to have such abundant protein as a target expression, so we should add such an artificial tag. Anyway, to have expression of target protein in host cell, we should go through vector, circular vector, and target sequence. So with such a recombination of sequence, we can add extra. Actually, it can be built-in vector, so we can easily obtain such a artificial protein with his tag from a target sequence. So his tag is very useful tagging for protein crystallography. Without his tag, Arne is saying we cannot study such a low abundant proteins. Most target proteins in drug development is low abundance. So to obtain low abundance protein in high abundance, the only way is overexpression in host cell with his tag. So that is his tag. And here, let's suppose our target protein and in sequence information, then we may have N-terminal and C-terminal. It's sequence, it's linear information. So to design construct, we can analyze some series of proteins, kind of family proteins. Among them, we can have some idea which part we need to express. So from the full sequence, we can decide which domain we would like to study. Even we can study whole proteins, but with some design. So we can, to develop such an idea, usually we are doing sequence comparison. So at the end of sequence comparison, we can decide which portion, N-terminal or C-terminal, we can add his tag. We are doing such an analysis at the beginning. Then crystallization. Crystallization, as we were talking about, protein may be not expressed, then we should obtain the construct which can be expressed. Then for expressed protein, we should obtain soluble protein. Then we should obtain crystallizable protein. But crystallization problem is not always guaranteed. For DNA sequence, we can synthesize DNA material, but protein, we cannot synthesize protein out of protein sequence. So even expression is not easy, even to obtain soluble protein is not easy, then obtain crystallizable protein is most difficult part. So what can we do for, let's have some break. Would you find the software or these pages from the textbook? 151. 151. External predict server, pred server. So Muhammad, for your sequence, try this server. Figure 4.5, prediction of crystallization success. You input your own sequence and you will get result like this. Okay. 다시 시작하겠습니다. 네. Let's start again. In now, from now on, we will talk about crystallization prediction. Unlike DNA sequence, protein synthesis or protein purification, even cloning, is not guaranteed. Rarely, we are talking about peptide synthesis, but we are not talking about protein synthesis. Why? Muhammad?
Because synthesis is of DNA. So
we can synthesize peptide. Peptide is part of protein. But we are talking about peptide synthesis, but we are not talking about protein synthesis. Why is the case?
because there are a lot of number of amino acids to synthesize protein, which is a very long, very large.
Very wrong. Firstly, very wrong to manipulate. But think if it is short, then you can synthesize protein of very short size, like brazane or.
I think proteins are expressed and purified. Synthesized is the DNA.
But we can synthesize peptide. Why not protein?
Peptides are combined in protein.
This is kind of open question, not a correct or wrong answer. Anyway, I'm challenging you in terms of your vocabulary. At the beginning, for Muhammad, I will repeat this again. This reminds me the time when Mui had troubles with her colleagues for practical molecular biology at that time. We have problem. What problem do you have? Which problem do you have? And why we cannot talk about and how we can talk about? I asked you why we cannot talk about protein synthesis while we can talk about peptide synthesis. And a peptide is a part of protein. So first thinking is like this. We should be able to synthesize protein of small size. But why not? Or how it can be possible? How possible? If not possible, why not? And if possible, how can we do that? Firstly, sequence, we are talking about model and science of Y and engineered of. In terms of Y of science, it should be possible. I'm giving you quick answer. But in terms of practice or engineering, it may be very difficult. Here we are talking about peptide synthesis and protein synthesis. Peptide versus protein. If peptide is possible, combining peptides, we can have protein. So it should be possible, first of all. So with the model of molecular biology, sequence from structure of proteins, sequence of proteins and structure of proteins. So DNA sequence and protein structure. DNA sequence to protein sequence, then protein structure. So what we are actually doing is, can we convert DNA sequence into protein structure? Why does it matter? Structure is improperly folded peptides are not functioning protein. What I'm trying, or what you should try, is like this. You should be able to express the problems you have. A problem of novice or beginner, I'm talking about non-expert, cannot describe the problem at the end, at the hand. In your hands, you have problem. Even it can be personal problems, interpersonal problems. You may have problems with your colleagues or fellow students. You are very challenged, or you are very in trouble. But with your boss or with your own professor, you cannot describe the situation. You will be under pressure, or you will be very challenged, but you cannot, because you cannot express the problem, so you cannot solve the problem. Not only in personally difficult situations, but also in technically challenging situations. You are doing your own experiment. Your boss has some background knowledge in that particular technology or experiment, but he may not have hands-on experience of particular machine or particular instrument or particular materials. You have better knowledge about particular situation or specific instrumentation, but you cannot describe the problem you have. Then you cannot solve the problem with the help of expert like your boss or professor. So, to be an expert, simply to be a master level of scientist or PhD level of scientist, you should be able to express the problem at your hand. That is the art of articulation, language articulation. You should divide, or you should analyze the problem at your hand, and you should use proper vocabulary which can be understood by an expert like your boss. He has some vocabulary, technical vocabulary. Using such a technical vocabulary and with your own common sense, you should be able to describe the situation, and you should be able to get help from him or her. That's the very essence I'd like to share with you in terms of coursework. General knowledge and textbook knowledge can be obtained through your own reading, but this kind of higher level of intelligence cannot be easily achieved. So you are in trouble in many technical challenging situations and many not personally challenging situations as you may have. So that is the point. And I challenged you with the issue of peptide synthesis versus protein synthesis. You may be told about peptide synthesis, but never protein synthesis. And I already told you, theoretically, it should be possible. No physics law hinders protein synthesis because already we have peptide synthesis. But answer is, technically or in terms of engineering practice, it may not be easy because of transferring sequence to structure cannot be automated. We may have full sequence peptide as synthesized. So let me ask this question. If you have same sequence, but same sequence, but different structure, simply a different folding, can it be functional protein? No. So to have functional protein or simply protein, what we mean by protein is functional peptide with some specific structure. So we may be able to synthesize some full-length protein sequence. Let's say full-length peptides of full-length protein, but it may not be fully functional protein. That is distinction. So from sequence to obtain structure is not in automation. Because of that, we are doing protein crystallography. If it is in automation, even given sequence, we can predict full structure. But from sequence to structure is not for automation. It's done in nature, but knowing them is not in automation. Nature is doing using what? From sequence to structure, how it is feasible in nature?
Transcription.
The simple word is translation. But translation is a little bit tricky, and I would like to give you direct answer as my own. Cell is doing translation, but not just full-length peptides, but also in correct structure. So cell is not only translating, but also guarantee fully functional structure. So cell is machinery behind nature. So cell, we were talking about host cell, but to have fully functional cell, we E. coli, mostly we have E. coli, but E. coli may not have enough machinery to have fully functional protein for signaling protein in high-level organism like human. Human cells may contain proteins which can be expressed in lower organisms like E. coli, but if such a protein needs to be phosphorylated, E. coli is lacking such a phosphorylation mechanism, especially protein tyrosine phosphatase and protein kinase, protein tyrosine phosphatase and protein tyrosine kinases. E. coli is lacking tyrosine phosphorylation and tyrosine phosphatase, but these two are very important protein family in human proteomics because they are implicated in cancer. Cancer is a disease which has problem in signaling, cell signaling. So these kinds of signal proteins may need higher organisms like human cell or even yeast cell, but E. coli may not be produce these kind of target protein. Anyway, from sequence to structure is not for automation. That's the whole purpose why we need protein crystallography to study structure starting from sequence. Also, we are talking about peptide synthesis and protein synthesis. Theoretically, protein synthesis is possible, but practically it's not possible because of difficulty in terms of folding. So cell is guaranteeing folding, proper folding, as well as proper modification. Did you hear about post-translational modification, in shorthand PTM? What is PTM?
Post-translation modification. For example, after translation, we can methylation.
Oh, for example, methylation. Yes.
Methylation, and we get the modified protein.
So, functional protein may need both proper folding of peptide sequence and to be functional in terms of signaling molecule or in terms of enzymatic protein. It needs modification. For example, methylation or tyrosine phosphorylation in PTP and PTK.
Kinase. K is a kinase.
Kinase, yes. Kinase. Tyrosine, TYR, amino acid. Tyrosine can be phosphorylated or dephosphorylated. Kinase is phosphorylating tyrosine site, so-called protein tyrosine kinase. And it can be dephosphorylated with PTP. That's the protein which Dr. Gang together with Sang-wook have studied. So this for proteomic study in human cancer, and they are kind of target protein for such a study. Anyway, when you're challenged with situation or problem, you should raise up. I'm saying raise me up in higher level. Which model we are talking about and why and how. Not just space-time and man-matter issue. You should think about these three questions, especially which model we are talking about. Then why possible and why not possible. Then how it can be or how it cannot be. So playing with these three words, why, how, which, you can analyze problem at your hand. This is kind of metaphysical questions. Metaphysical, above physical event. Which is kind of a word you can use for any kind of situation, like which person, which matter, which time, even which place. So kind of generic word, so it's not part of 5W1H. 5W1H consists of why, how, and who, where, what, when. Particularly who, where, what, when is kind of physical word, even including who. In terms of science, language is the most important tool in science, right? Simply research. We learned through the textbook recently, right? But in terms of science language, man is observer, not only subject but observer. In physics, you know the name of most famous physicist Einstein. And at the time of Einstein, we discovered some Einstein relativity theory, and at the same time of 20th century, we discovered quantum physics. Coupled with Einstein's work on relativity, famous equation like this, E equal mc square. With the advancement of quantum physics, we discovered secret of atoms and atomic bombs. Then whole physics around subparticles like particles below electrons were discovered, and we are talking about uncertainty principles. And in terms of philosophy, now we are going to philosophy of science. Always we have observer and event. So observer, and without observer, we cannot describe the event. So observer is a kind of formally, what I'm trying to say is this is kind of beyond this topic, but let me finish this quickly. Unless you are comfortable with physics, ignore whole, but remember this. Which, why, how are metaphysical questions. Especially when you are in trouble, think about your problems in terms of these three. Normally, people are concerned about know-how, which is question of how, of technique. But also think about why and which model we need to describe the problem. And physical world. Physical question is like this: who, observer, and subject, and what, and where, and when. These are space-time issue of whole physical event. These are man and matter issue. Materialism is like this. We cannot separate man from matter because man is a kind of matter. Man is made of, I'm not specifically talking about gentlemen, I'm also including ladies like you. So from the result of modern physics, we realized man cannot be separated from matter. Observer cannot be separated from event. That's the point I'd like to share with you. So, in all ancient times, we thought man is special. Who is special? So, who before the modern physics? So, physics and philosophy. Before the modern physics, we have naive idea about our own identity, and we have tendencies to separate who from the other three. But after modern physics, observer should be part of a system. So observer still part of physical world. We are in physical world anyway. So who, what, where, when can be analyzed in material world or simply physical world. So with these 7 words, you can analyze whole things. Any kind of troubles. That's the point I'd like to share with you. And this is kind of extra thing at the level of master's or PhD. Using these keywords, you will never miss the essence. So this is kind of weapon, language weapon for you. Okay? Anyway, let me go back to our original problem and crystallization. I was talking about expression and expressible protein and soluble protein and crystallizable protein. There are 3 levels. Even we have non-expressive protein, even we have non-soluble, simply insoluble protein, and we have non-crystallizable protein. So crystallizable protein is rarest one. It's not common one. So by having overexpression of protein, it does not guarantee we have crystallizable protein. So once we try overexpression with recombinant DNA technique, we should check whether our protein sequence can be crystallizable or not. Also, even if we have some known packed or similar proteins which were crystallized, such a pack doesn't guarantee our target protein can be crystallized. It's not guaranteed. So at first level, prediction. Second level, actual expression and crystallization experiment. Both should be done to avoid some problems beforehand, not jumping into actual crystallization. I told you crystallized proteins are rarest one, most infrequent one. So not all proteins are crystallizable. To avoid some failure through expensive expression and crystallization experiment, it's better to check using prediction server, as I told you, Xtal PLED server. Then your protein can be very hard to be crystallized. Then you should modify sequence beforehand. Even without relying on such a server, by being an expert, you can have some intelligent guess. Particularly, transmembrane protein cannot be easily crystallized. You should cut some insoluble part beforehand. Then you can obtain some soluble domain or soluble part. Likewise, you can do analysis beforehand and you can avoid some difficult cases, or you can focus on soluble portion of whole protein. Okay, that's the point I'd like to share with you in terms of practice. And the last sentence is like this: crystallization trials remain the only authoritative and in fact fast and simple method determining crystallization probability of target protein construct. But this can cost a lot, so to avoid that, for your own sequence, it's better to check in advance using some software or relying on some expert advice. This is software screen capture. I advise Muhammad to write this one, but his case, if he tried to have some recombinant one based on existing one, we may have some proteins which are already crystallized. Then just changing one or two amino acids will not change the situation a lot. So we can promptly proceed actual crystallization anyway. Here you can find some consequences, single amino acid changes. That's the topic we are mostly interested in. Recently, for red fluorescent proteins and cyan fluorescent proteins, what we are doing is single site amino acid changes. So through such a series of mutation, we direct modifying existing one with new properties. What properties do you expect?
Change of color.
Your case, change of color, but I may not want change of color because already we have broad spectrum of color changes. You may be interested in other property.
Abundance.
Mm-hmm.
Abundance.
Even expression level can be changed. But personally, I'm more interested in crystal size or crystallizability. Let's skip this part. So for today's topic is proteins for crystallography, and for crystallography we should have crystal. Without crystal we cannot do crystallography. So actually the topic is proteins for crystallization. The way to increase crystallizability, especially we have similar proteins which were already crystallized. I'm talking about some proteins in family. For most structural papers or structural report, we have sequence comparison like this, amino acid sequence comparison, our target protein and their relatives among reported one or from database, especially of their structures were already reported, then we can make some comparison and we can have some guided guess like domain structure and the arrangement. From them, we can increase crystallizability of our carrier or target protein using single site amino acid. That is the topic discussed here. Selection of residue, simply amino acid for surface entropy reduction. Some proteins and some amino acid have very flexible side chain. From chapter 2, we talked about several amino acids like lysine or arginine. They are lengthy and flexible. So by changing such a flexible amino acid into compact one, like which is most compact one in terms of size of side chain? Alanine is compact, but we have more simple one. Alanine has methyl CH3. Glycine, yes, bingo. But alanine here mentioned for surface entropy reduction because it can mimic some other amino acid with C alpha. Glycine is lacking C alpha carbon. So by maintaining C alpha, alanine can be replaced easily without disrupting too much. But glycine is too small, so I don't know why glycine is not the case, but it can be such case. So rather than having flexible side chain, if we replace the amino acid by alanine, still alanine can mimic C alpha chain, but lacking flexible side chain, it can be easily, it can increase the chance of crystallizability. That is the technique so-called surface entropy reduction. Anyway, do you know the word entropy? Can you describe entropy?
Change in the original.
You know the word energy. How entropy is related with energy? I'm checking your physics background. Anyway, let me read this and.
Increase of entropy causes the decrease of system energy.
So, briefly, entropy to a proper knowledge, energy and entropy are related. Energy and entropy. In terms of dimension, energy and entropy do not have same dimension. We need a kind of conversion factor here. Entropy usually written as S, energy written as E, and what should we have here? Kind of quiz, quick quiz. Very close in terms of symbol, but T is not time.
Time period.
Huh?
Time period.
Muhammad said time period of time. But this is not period of time. So, what else do we have in physics with capital letter T? Even torque. Honoring your all answers, I will give you promptly correct answer. This is correct or wrong questions. Here, correct answer is temperature. don't blame yourself. This is simply you are in different worldview. You are from biology or veterinary medicine, veterinary science, not from physics, so it's okay. But now, don't forget temperature with your entropy. Why I'm talking about this has something with later part. Crystallography is always talking about temperature vector. I'm saying temperature vector. And simply, the word you should connect with entropy. This is also about vocabulary building. Crystallography is not only for structural biology, this is also from physics. So basic, you should understand basic model. I'm talking model. If you are missing some key idea, I will not try to teach you physics. But if you miss this kind of important link about some conceptual understanding, you cannot comprehend the logic behind them. Simply, you cannot understand the worldview. So, temperature vector in crystallography is related with temperature. I said temperature vector is related with temperature. Likewise, entropy is related to energy through temperature. And this is about degree of
change.
You said change, but better word is disorder. What disorder we may have in terms of increasing temperature, like vibration in atomic model. In crystallography, at X-ray room, we have some liquid nitrogen cooler. And if you look at the textbook, we are talking about cryocooling of protein crystals. By lowering temperature, we can reduce disorder, and disorder is related to energy term. So delta E, delta S, change in entropy is related to energy proportional to temperature. Higher temperature means higher energy changes with the given disorder. Anyway, with the word of entropy, you should connect the word disorder rather than change. Change is kind of neutral word, but disorder is not neutral, kind of bad thing. If order is good thing. Anyway, crystal is about having order over space. Regular thing. Disorder is kind of irregular thing. Irregular versus order. So, without understanding the word entropy properly, you never capture the meaning of entropy reduction here. So, what we are reducing here? Entropy reduction. Here, let's forget about surface. Let's focus on entropy reduction.
Disorder reduction.
Yes, we are reducing disorder. By having flexible side chain, we have great level of disorder. With many dihedral angles or torsion angles, flexible, then we have great disorder. With such a disorder, crystal is about ordering, so crystallization can be hindered. By reducing such a disorder, we can increase crystallizability. So we need to reduce disorder level and simply entropy level of entropy. Okay? So now you have an idea about entropy and the purpose of entropy reduction. But what about surface here? Why did we attach the word surface in front of entropy reduction? What surface are we talking about? Our face or crystal face or what face? Anyway, surface is face. Surface is face. Which face we are talking about? Which face? Face of crystal. Actually, we have terminology for face in crystal. External face of crystal. For example, you may have some jewelry. Diamond is most expensive jewelry. I'm talking about high quality diamond. It has face, external face. Actually, it is called face in crystallography. Even we have some numbering for each particular face, HKL face. But this face or surface is not such a face. This face is protein face, protein surface. Don't forget folding. To be functional protein, we should have folding. Once we have proper folding, protein may have interior part and exterior part. Exterior part can be called surface of protein. We have inner part and outer part. Outer part is called surface of protein, and amino acid or residues which are on such a surface should be mutated, because surface matters in terms of interacting with other proteins. I'm talking about same proteins which should be ordered into crystal to have regular packing. If you have flexible side chain exposed on surface, it can hinder such a regular packing. So we should mutate such a external or surface amino acid into simple one like alanine. Then crystallability will be increased. Muhammad, what you did in terms of mutation recently for your M cardinal 2, what was your primary mutation?
Alanine and glycine.
That is my favorite mutation.
Yes.
Because of this. Basically, I'm not a biophysicist. I'm a crystallographer. Also for neutron crystallography, which I didn't discuss with you, we need to have big crystal. Big crystal can be obtained with this kind of surface engineering to enhance crystallizability. That's the reason why I'm so interested in this kind of mutation. Okay, that was not for color change. That was not for what was LS shift?
Large stock shift.
Large stock. That was not for large stock shift. That was for enhancing crystallizability. Now you can understand what I'm talking about. Anyway, for you it may not be easy topic, but for not just for crystallography coursework, when you have trouble in terms of analyzing some problem, go back to model issue. Which model we are talking about? The answer you gave at the first time, time. It was not time. If you take this as time, you have wrong model. If you are talking about time period, because this was capital, you made intelligent guess. Usually time is written as simple small letter, time period T, but both are wrong. Both are in different world, simply wrong place. So talk is in physics, but it was not talk either. So you should have correct model. You do not need to understand whole physics behind this, but you should have proper language and proper model. Without having proper model or proper language, you cannot describe the problem at all. So your running objective in completely new field. Anyway, even for PhD scientists, time to time we need to work in other field which you may not be familiar with. Then your primary learning objective for such a new topic is like this: learn the vocabulary and develop your vocabulary to the level we can correctly describe the model. You do not need to understand whole details, but you should be at the level of describing the model properly. You should not make a mistake like this: temperature versus time, because temperature versus time are totally different physical level. That should be your lesson. And let me go back to crystallization. And now another important or most important lesson of today, protein crystallization anyway. But I added something under this main title. After studying this chapter 3 last time with Mui, I discovered some main idea was not described in the textbook properly. The main is like this: dewatering. What does dewatering mean to you, Muhammad? On behalf of two.
Removing the water.
Yes, removing the water. So, from your protein stock solution, what do you have in your case?
Buffer and precipitant.
Not yet precipitant. If you precipitant, you may not have good solubility and your stock solution in trouble. Only during protein crystallization screening, you may add precipitant. Think about your gel filtration bubble. Do you have precipitant there? You should not have precipitant there. If you have precipitant there, you may decrease solubility. As a result, you cannot make concentration. Okay? So, solvent here means two things. If you mix any, dewatering is removing water from the solvent, and I was talking about solvent in two different stages. One is purification. Let me talk about. So we will not remove. Actually, we are removing water. Not only removing water. This is worth to talk about. For protein concentration, at the final step of purification, what do you use, Muhammad?
Vivaspin filtration.
Yes, Vivaspin.
Filtration.
I'd like to compare these two things. Vivaspin, Vivaspin. What does Vivaspin do for you? Do you know something about Vivaspin? Do you know? Vivaspin is special filter to remove solvent out of protein solution, simply concentration technique. So you may have chance to look at Vivaspin while Muhammad is working. So this is kind of a technical word. So you may not need to know Vivaspin, but you may see Vivaspin while he is working. And what does Vivaspin for you?
Concentrating the protein, removing the solvent from protein.
So remove solvent. Vivaspin to remove solvent. But I was talking about dewatering solvent through crystallization. So we have crystallization remove solvent. But here, PBA spin and crystallization purpose, their purpose, like proteomics and protein crystallography, we have some similarity and differences. I'd like to talk about difference between PBA spin and crystallization. First of all, what do you have in your mind in terms of differences? Any common thing is concentration, higher concentration. Any crystal has higher concentration of protein. PBA spin will result in higher concentration of protein stock solution. Both are common in terms of blazing protein concentration in the cell or in the system. But what will be the difference between PBA spin and crystallization?
In crystallization, concentration is too much high. PBA spin cannot do like that.
PBA spin, if you exert too much, you may have some bad thing. What bad thing can you experience even with PBA spin?
Aggregation of protein.
Oh, very nice. Common thing is either crystallization or PBA spin, we can result in aggregation. Aggregation or nucleation both means insoluble. Any even soluble protein can result in insoluble if you raise its concentration too high, right? But crystallization and PBA spin has something common in terms of decreasing solvent amount from stock solution and increasing protein concentration. But I'll not talk about PBA spin from now, but we will talk about crystallization. But later then, you can discover difference between PBA spin and crystallization. Simply PBA spin, like a sieve. This is kind of exaggeration. We have much smaller scale. What is the size of your PBA spin?
10 kilodalton.
PBA spin has some specific molecular weight for sieving or simply filtering with the given. But what is the size of your protein?
29 kilodalton.
29 kilodalton. So this is sieving of this molecular weight. So what will happen if you put your protein solution here and apply? What do you apply in terms of centrifuge?
3000 RPM, 4 degrees centigrade. Time depends on until final concentration.
You are allowing time for concentration. You are centrifuging PBA spin with protein stock solution, and what you will have is kind of non-protein or any kind of solvent molecule or molecular weight is lower than this filtering size, right?
Yes.
Then here on top you have remaining protein. Okay.
Yes.
Correct? So any solvent of lower than this size can be transported on bottom, and protein will remain on top, right? This is the mechanism of PBA spin. But crystallization, we mostly using sitting drop or hanging drop depending on geometry for pictures. Here is a picture showing hanging drop and sitting drop. Last time we talked about sitting and hanging, but for Sabah, I'll briefly talk about difference between hanging and hanging. This is hanging drop. We don't have sitting drop. For example, this is hanging drop, but you may find the description of sitting in the text. But anyway, point is crystallization. We should have mixing. Here, Viva spin, we are dealing with single protein stock solution, but for crystallization, we are mixing two solutions to induce crystallization. Mixing protein stock as well as here there's a bubble or cocktail. They are talking about cocktail. Cocktail is textbook word, but better word is a kind of precipitant solution. Stock solution, protein solution, and precipitant solution, which is called cocktail here. But the reason why it is called cocktail is we may have buffer solution as well as some precipitant molecule like a polyethylene glycol or some salt to reduce which parameter? Precipitant is to reduce some parameter. What is the parameter? Simple word starting with S. Yes, solubility. Yes. Here, Viva spin is simply remove solvent to the highest possible solubility. It should not result in aggregation. Okay, it should remain within the range of soluble. But here, this is opposite. With the given soluble protein, we should induce insoluble or nucleation. Better part is nucleation. By mixing, starting with single pure protein solution, we are increasing concentration up to the level of just below the solubility limit. This should be under solubility limit. Concentration, better word is concentration. Viva spin is technical word and generic word is concentration. Concentrating of protein. Crystallization of protein. Both are two processes in opposite direction. To do protein crystallization experiment, we should have concentrated protein stock solution. But through concentration, we should stay under solubility limit. But to do crystallization, we should exceed above solubility limit. Once we exceeded solubility limit, as a result, we should have some insoluble part. But by being harsh or rush, such an insolubility will result aggregation, as we might have aggregation at the concentration stages. But crystallization, we need gentle treatment, which will lead in crystal rather than aggregation. Aggregation is an irregular assembly. Crystal is regular assembly. So crystallization should exceed some solubility limit, but page diagram. Here, solubility. By playing precipitant, by mixing protein and precipitant, we are going through this way. This way, and this region is good for aggregation as a nucleation, but this region is aggregation, disordered aggregation. Ordered aggregation is called nucleation, and disordered aggregation is called simply aggregation or precipitation. So here we have figure for sitting drop and hanging drop, and you can follow the description. And here, this phase, by mixing protein stock solution and precipitant solution, we may have reduced concentration because volume is increased by adding 2 drops. This drop, protein, and this drop from precipitant or reservoir. In terms of location, they are in abundant amount. So because of such abundant amount, for example, this can be 1 microliter level, this can be 100 microliter level. So because of relatively larger amount of this solution, it can be called reservoir solution. But in terms of its function, it can be called precipitant solution. So by using different words like torsion angle and dihedral angle, we can designate same thing but with different meaning or different roles. Anyway, by mixing precipitant solution and protein solution, we have larger volume than original. Each volume can be doubled if you use 1 microliter of protein and 1 microliter of precipitant. Total amount can be 2 microliter. So here, because volume doubled, we have half concentration protein and half concentration precipitant. So in phase diagram, in terms of protein concentration and precipitant concentration, from the original is here, but mixing one here. So initially, by mixing these 2 drops, it can be inverted, and in terms of geometry, it is called hanged drop, and after sealing, reservoir and hanging drop can be in equilibrium. Then what is happening after initial setup?
Vapor diffusion.
So-called vapor diffusion. But what molecules can be transferred through such a vapor diffusion? Vapor means gas phase diffusion.
Solvent molecules.
What solvent molecule is transferred?
Water or buffer.
That is tricky part. So I talked about solvent, but we need to understand model behind this. Okay, so I'll not give you answer, but especially for Muhammad, you need to specifically answer whether water alone or even including some salt or PEG can be transferred. Okay? So here, if I give you for the other two, Mui and Saba, only water in this model. I'm talking about model now. Any concentration, any molecule of which size is smaller than filter size. Previously, you were talking 10 kilodalton. Any molecule, solvent molecule, smaller than 10 kilodalton can be removed. Only protein of which size is larger than such a filter size will remain. But here, vapor diffusion crystallization experiment is not like concentration, only water. I'm giving you answer. Water is transferred. You can think about why water is transferred in that model, but I'd like to remind you, model is model. Model. Recently we talked about theory and truth. We have difference between truth and theory. Theory cannot be proved to be either true or false. It can be supported, right? Truth can be proved, but theory can be supported. Likewise, model is about approximation. in reality, through vapor diffusion, even small molecule, like let's suppose we have some ethanol there. What did you have your M cardinal 2 crystallization?
As a buffer?
Yes, buffer or solvent. Precipitant.
PEG and calcium chloride.
And you had something else, MES and...
Yeah, MES. MES and NOS.
And didn't you have methanol or ethanol there?
That was in other composition.
In other composition. Anyway, water, HOH, H2O, and methanol and ethanol was volatile. So molecules like this can be volatile and it can be transported through vapor diffusion. But most times we do not have methanol, ethanol in our buffer, so most times volatile molecules will be water. Water is volatile, which can be transferred through vapor diffusion. So if you look at phase diagram and if you're concerned about computation of concentration, you may realize only water is transferred in this model. I'm talking about model. So in reality, we may have very little chance, but still real chance to transfer other molecules. But in this diagram, in theory, only it deals with water. That is my point of view in terms of understanding model. So you can actually calculate what is starting concentration and what is resulting concentration or final concentration with this model. So without understanding what molecules can be transferred, you may not be able to calculate this diagram. So secret about this diagram is to understand which molecules are actually transferred in this model. This can be beyond your scope. I'm sorry, Sabah, but anyway, this is kind of effort to understand model behind this kind of solubility diagram. And in this textbook, we had several diagrams like this to describe some particular phenomena during vapor diffusion experiment. But Muhammad, you actually need to understand the logic behind this. Here, you didn't use 1:1 ratio.
Yeah,
Which ratio did you use?
3:1 ratio.
3:1. Then, even with the same concentration, your starting will not be here. This is only for 1:1 mixing. Serial mixing, you can calculate 3:1 by dividing volume against amount. You should be able to manipulate. Then you will have different result. From the textbook, you may find that information, but for others, it's beyond some your own interest. So, as not as a student, but as an expert, you should be bold to skip some topics you are not interested in. So you can safely ignore this topic because you will not exercise crystallization in practice. But Muhammad is actually doing crystallization even for his own setup. He is not only using 1:1 ratio of mixing, but he's also trying to another or different ratio. So to have estimation on this phase diagram, if we mix in different ratio, this arrow will not in 1:1. This can be this or this can be here. So end result will not be the same. So you need to understand the logic behind this, and the hidden logic is only water is treated as volatile molecule. So considering protein concentration and precipitin concentration together with the volume you are adding, you can calculate final volume and final concentration. Okay, so you can skip. So anyway, and the results should be like this. This is kind of colorful gallery of your crystallization effort out of your crystallization trial. Muhammad may get some result like this. This drop is kind of microscope view. Here, this boundary is part of such a drop. Usually, we are adding 1 microliter. So initially, it can be 2 microliter drop. So this size cannot be larger than 1 or 2 millimeter in diameter. So this should be smaller than several 100 micrometers. Actually, it's around 100 micrometer. But here, a palm is like a needle, and here and there, mostly aggregate. So we should try to obtain something like number 1 or 3. But in most times, we can have needle form like that. So we should manipulate precipitant condition. Also, we should change a mixing ratio. So even we can change buffer condition. So to change buffer condition, we should do screening kit with many varieties of buffer. So once we have aggregation, it means we have solubility problem, or not solubility problem, high aggregation problem. So we should apply more gentle condition, which can result in nucleation. So anyway, by making observation like this, you should plan your crystallization accordingly. Any question? Muhammad,
Yes.
here, this is like your sitting drop condition, and here reservoir, tail plate, and what you are looking at in terms of arrow,
Vapor diffusion.
vapor diffusion. Here, same vapor diffusion in slightly different dimension. Basically, sitting drop and hanging drop, as you might notice, in principle they are the same. Only geometries are different. This hang on top, the other is sitting on different location. Okay? Sitting and hanging. But this green one, as I told you, they are transferring waters from here to reservoir. Initially, we have too much water there. So by transferring water from mixed drop to there, what will happen in terms of volume? Decreasing. But it may depend on time. In terms of real practice, we need to allow time for crystallization, but if it is taking forever, it will not be practical. So when you use your different mixing ratio, in theory it's identical, but it may be identical in terms of crystallization time. With a given protein stock solution, by compensating protein stock, in the original paper they used, I'm specifically talking with Muhammad to help him, but for you, you may not need to understand whole about this, but in practice, this does matter in terms of time. I was talking about 4 physical words: what, who, what, man, matter issue, and when and where, space and time issue. Anyway, this is about space, transferring through space, but we also have time differences due to different volume ratio. In terms of protein concentration, we can have, for example, 10 milligram per ml as a starting or
20 milligram
stock solution. Anyway, achieving higher concentration can be risky because of aggregation during devaspin or protein concentration. So it may be safe to play under 10 milligram per ml. But to induce nucleation, having higher protein concentration is better. But to compensate of low concentration of protein stock solution, we can apply more volume of protein drop, as you did. Rather than 1 microliter, you can apply 3 microliter. For example, if you had 30 milligram per ml for your own experiment, you can use 1:1. But they had 10 milligram per ml, so to compensate low concentration of protein stock solution, they should exert triple amount of volume. Then final amount will be the same. But initial volume will be increased if you use higher volume or larger volume of protein stock solution. So in terms of protein amount can be same, but starting volume will not be the same. So in practice, it will take longer time for dewatering. I'm talking about time aspect of your crystallization experiment. So not only space, but also time matter in your experiment. So equivalent in theory may not be equivalent in practice. We human beings have limited time. It's better to know result in short time rather than in longer time period. So you should consider that aspect too. Crystallization can be observed within 3 days. But low concentration protein stock solution, you should apply larger volume. Dewatering of larger volume will take longer time. In extreme case, it will take several months. In real cases, for example, very difficult to crystallize because protein is not very soluble. Also, it is not very compact. Then it will be difficult to be crystallized. Then such a crystallization can take several months. After several months, we can have very handful amount of crystals. That is possible scenario, but you may not want such a case, Muhammad, when you can crystallize your protein within 3 days. So you should be careful about your starting concentration, and you should design your volume for setup. Anyway, I'm done. Question? I'm sorry about this technical discussion, but for you, you can ignore some technical details, but the point I talked with Muhammad is time aspect of experiment. Even for your own experiment, for chlorella cell culture, some practical problems, you can think in terms of not only space, but also time. Even for cell, anyway, you have cell compartment. To extract some part and some target protein, you may need to fractionate your sample using centrifuge. Then I don't know much about chlorella, but for example, chlorella has chloroplast for chlorophyll. For some proteins associated with chloroplast, it's better to fractionate chloroplast part using some analytical or preparative centrifuge. Then it's better, it will give you better chance to work with such a target protein. And separate in time and separate in thinking in terms of time and space may give you an answer about some difficult or challenging technical problem. That's the point I'd like to share with you. Muhammad, you think in terms of not only in terms of geometry but also in terms of practical time. You may not want to spend several months when you have deadline of several weeks. Okay. We talked about culture problem too. For next chapter, I will develop some quiz together with some video material. And we finished some protein part, and we will come back to crystallography again. But crystallography, and mainly vocabulary building to understand your case, to understand others' research. We will not go deeper than the vocabulary well for you. But for you, Muhammad, you need to expand your practical knowledge, not only in terms of basic vocabulary, but also in terms of practical terms. As we discussed today, kind of timing consideration. Okay? If your experiment takes forever, it's not practical. We should make sure we can finish the experiment in time.